Sorry, you guys. I was interrupted by my beagle who decided it was breakfast time. I haven't quite figured out how to pause or stop a QuickTime screen recording as I'm making it, but I am working on it. Thank you for your patience. This is, we'll finish off lecture 25 with this small add-on to the previous file. In any case, we left off at chromophore regeneration. So the key takeaway is once 11-cis retinol has photoisomerized to all trans retinol, it needs to be refreshed, right? So all trans retinol will unbind from opsin, and 11-cis retinol needs to bind again. So the chromophore needs to be recycled. It needs to be converted back from all trans into 11-cis. And that process takes place in two different places. It takes place in the RPE, the retinal pigment epithelium, layer 1, which we said is important because it can phagocytose the ever-extending tips of the outer segments, and is important because it contains melanin and it reduces the scatter of light within and around the retina. A third important function of the RPE is chromophore regeneration for both rods and cones. Mueller cells, if you go back and look at the different layers of the retina, you'll find the Mueller cells, and they are important for regeneration of 11-cis retinol, specifically for cones. So let's start with the RPE, and let me preface by saying there's a lot of biochemistry on this slide, and I would never ask you to reproduce this diagram, but you do need to understand the basics of the interconversions that are happening, where this is happening, and maybe some key differences between the biochemical reactions in the RPE cells versus the Mueller cells. Okay, so let's start with we're in a rod or a cone cell. We've photoisomerized, so we have all trans retinal. The first interconversion that takes place is all trans retinal, the aldehyde, to all trans retinol, the alcohol. And all trans retinol will be transported from the rod or cone photoreceptor to the RPE. And that's done by IRBP. I think I forgot to put that one down here at the bottom for you, but that's intracellular retinal binding protein. Okay, so it's a retinal binding protein, RPB, that carries the all trans retinol from one cell, the photoreceptor, to another cell, the RPE, a retinal pigment epithelial cell. So IRBP is intracellular retinal binding protein. So now we've got all trans retinol, the alcohol, in the RPE, and there will be a number of esterification and isomerization reactions that will ultimately get us 11-cis retinol, the alcohol. And that's almost what we need, but we don't need 11-cis retinol, the alcohol. We need 11-cis retinal, the aldehyde. Okay, so RPE cells express 11-cis RDH, where RDH is retinol dehydrogenase, which will do the conversion from the alcohol to the aldehyde. And now we've got what we need. Okay, and that will be transported. 11-cis retinal is regenerated in the RPE, and it'll be transported by the cellular retinaldehyde binding protein, or CrALBP, via intracellular retinal binding protein back into the rod. And once we've got 11-cis retinal back, we've got our photosensitive photopigment back, and the phototransduction cascade that we talked about on previous slides can begin again. Okay, so that's the pathway in the RPE. In Mueller cells, things are very similar, except the Mueller cell doesn't do the final conversion. The Mueller cell, and you can put one additional circle here on 11-cis retinol, that's the alcohol. So that's almost but not quite what we need. So the Mueller cell will do all of the conversions to get us back to 11-cis retinol. However, the final conversion from the retinol, 11-cis retinol, to 11-cis retinal, the aldehyde, which is what we want, is done by the cone cells. So one difference between rods and cones is that cones, but not rods, express 11-cis RDH, retinol dehydrogenase. So cone cells can do that final conversion from the alcohol to the aldehyde all by themselves, whereas rods cannot. One difference between Mueller cells and RPE cells is that RPE cells express 11-cis RDH, retinol dehydrogenase, whereas Mueller cells do not. So Mueller cells can't make that final interconversion. However, RPE cells can. Okay, so a couple of key differences between RPE cells, Mueller cells, and a couple of key differences between rods and cones. I would never ask you to draw out all of the biochemistry on this slide, but if there's a circle around it, you should be familiar with the compound, and you should know what's happening where in terms of overall differences between rods, cones, and RPE and Mueller cells. And key points are summarized over here for you on the right with bullet points. Okay, so then in terms of basic circuitry, this should now be review. I've highlighted the direction of light, the pathway for light, and it is opposite to in the opposite direction of the signaling pathway. Light bypasses the ganglion cells, it bypasses the bipolar cells, and then finally it hits the photoreceptors. And again, I'd like you to think about why that might be the case in terms of high acuity vision. And then the signal transduction goes in the opposite direction in that it's initiated in the photoreceptors, which will decrease the release of neurotransmitter glutamate in response to light. And that will signal bipolar cells, horizontal cells, amacrine cells, and ultimately the retinal ganglion cells, which are the only neurons here that fire action potentials. Actually, amacrine cells can fire action potentials too, but the physiological importance of that is unknown. So for our purposes, we're not going to worry about that. We said that the horizontal cells and amacrine cells are interneurons. They're important for lateral inhibition. Put a circle around the horizontal cells because they're going to be important in coming lectures. But don't worry about the amacrine cells. You know that they're there. You know that they can fire action potentials. You know that they are interneurons, but you don't need to worry about them any more than that. Okay, so key cells, the photoreceptors, the bipolar cells, the horizontal cells, and the retinal ganglion cells are the ones we're going to talk about in depth. Key points: photoreceptors in the dark are depolarized. That is always true. Photoreceptors in the dark are depolarized, and they're releasing glutamate onto bipolar cells. And next time we're going to talk about two different types of bipolar cells. So glutamate release in the dark, remember, it's not about the neurotransmitter. It's about, and if we were in lecture, I would ask you, it's not about the neurotransmitter, it's about the receptor. It's about the receptor. And there are glutamate receptors, as we've seen already, that can be excitatory, and there are glutamate receptors that can be inhibitory. And so some bipolar cells are inhibited by glutamate release, and some photoreceptors are excited by glutamate release, depending on the type of receptor that they express. So in the light, glutamate release is, and here I would ask you to fill in this blank, in the light, glutamate release by photoreceptors is increased or decreased. And hopefully you'll come up with decreased. In the light, glutamate release is decreased, which excites some bipolar cells, and bipolar cells, when they're excited, in turn release glutamate, neurotransmitter, onto the ganglion cells, which excites the ganglion cells and increases their rate of firing. So it's a little more complicated than that because there are two different types of bipolar cells. On bipolar cells, which are turned on by the light, they're excited by the light, and there are off bipolar cells that are turned off, that are inhibited by the light. And their response to glutamate release or decreased glutamate release by the photoreceptors, because it's always true that a photoreceptor will hyperpolarize in the light and decrease glutamate release in the light. That is always true. But the response of the bipolar cell will be determined by the type of glutamate receptor that bipolar cell expresses. Okay, so next time we'll talk about on bipolar cells and off bipolar cells. Let's just finish off with a quick summary of of opsins. Opsins are GPCRs. An apoprotein it's a protein that needs something else. So it's almost but not quite a completely functional entity. So you would correctly refer to opsin as an apoprotein. And then the photopigment is the apoprotein opsin plus retinol, which is 11-cis retinol. I've said that these are the best studied of all GPCRs. So there's a number of different crystal structures that have been solved using X-ray crystallography. The best is bovine or cow rhodopsin. And we know from that and other structures that there's 7 transmembrane spanning helices. I highlighted earlier there's an extracellular N-terminus and an intracellular C-terminus and intracellular G protein binding domain. All opsins, in order to be part of the club, the GPCR club that is an opsin, right? All opsins are GPCRs, but all GPCRs are certainly not opsins. In order to be an opsin, you need to have a lysine residue at amino acid residue 296, and that's by bovine rhodopsin numbering. So it's not always exactly residue 296. It might be 295 or 292 or 301, right? But somewhere around this region in helix 7, there is a critical lysine residue that we refer to generally as K296. And K296 is important because that's the chromophore binding site. That is where 11-cis retinol binds. You have K296, you can join the opsin club. You don't have K296, you might be some other kind of GPCR, but you are not an opsin. Okay? And then a question that often comes up is, what about the differences in spectral sensitivity for 420 versus 500 versus some other wavelength? And that's due to differences in amino acid sequence, and we don't care exactly which amino acid sequences vary between the short wavelength and medium wavelength and long wavelength sensitive opsins, just that they do vary, and that changes the chemical structure and the chemical nature of the retinol binding or the area around the retinol binding site, which will in turn determine which specific wavelengths of light will activate that particular opsin. So suffice to say that differences in spectral sensitivity are due to differences in amino acid sequence. And down here at the bottom, you can see the critical lysine residue, K296, binding to the, in this case, it's all trans retinol. And for those of you that are more chemically minded, that particular chemical binding is due to or as a result of a shift base. But we won't go into the chemistry of this interaction. Suffice to say that K296 is a critical residue in order for GPCRs to join the opsin club. All right, I'll leave you with some review questions. I hope you enjoy your weekend to the greatest degree that you can. Please continue to follow health advice and stay in and stay well. And I will talk to you on Monday.